Hello, I'm Karsten Bonham, and I'm a pediatric neurologist, physician scientist at the National Institute for Neurological Disorders and Stroke. And I direct a section on neuromuscular and neurogenetic disorders of childhood here in the neurogenetic branch at NIH. I've been at NIH for 10 years now. Before that, I was at Children's Hospital of Philadelphia. Before that, at MGH and Boston Children's Hospital with a detour in Germany. Here at the NIH, we are mostly working on early onset neuromuscular and neurogenetic conditions affecting children. All of them are genetic in nature and by their very nature, rare diseases. So rare disease research is what we do on a day-to-day basis for children with neuromuscular disorders.
Hi, everybody. My name is Chuck Vendity. I'm a senior investigator in the National Human Genome Research Institute and the director of the Organic Acid Research Section. I'm a pediatric geneticist, and my focus area is on the treatment of children with inborn errors of metabolism. And at the NIH, I've been studying a group of organic acidemias called the hereditary methylmalonic acidemias and propionic acidemia, which are grave conditions of mainly infancy and childhood. And for me, my focus on this area began when I was a fellow at the Children's Hospital of Philadelphia. And interestingly, in those days, Karsten and I sort of overlapped. He was an attending physician and I was a fellow in a different department in genetics. But newborn screening, and for those who don't know what this is, this would be when the baby gets a blood taken from them in the very first or second day of life to test for a variety of conditions. And in the late 1990s, there was a movement to test all babies for hereditary methylmalonic acidemia and propionic acidemia syndromes. And that is when I first met families who had babies that had these conditions that were posing the question to me of what is going to happen to my baby? Is there a treatment for my baby? And so that, for me, working with the parents, I began a path towards developing new animal models and treatments for that group of disorders, which I continue to work on this day in the intramural research program here at the NIH.
Yeah, my story really started as a medical student in Germany. In my first year of medical school, in Germany, we had to do a nursing rotation, and I did it in a pediatric neurology setting on a ward for children with pediatric neurological disorders. And that really did it for me. After that, I only wanted to be a pediatric neurologist and find out what was happening to these children, what was causing these disorders. And that got us into neurogenetics very quickly. The research we are doing at the NIH is to try to understand the genetic causation, the mutations, the genes affecting these children, but now more and more also to find therapies that are gene-directed, that go to the source of the problem, including gene therapy.
Well, I feel very fortunate to be a member of the PaVe-GT team with you, Karsten, and your associates. And I'm very grateful for the excellent team that we have assembled here in the NHGRI, including excellent and skilled laboratorian colleagues with decades of experience in gene therapy. And that would be my colleague, Randy Chandler, a senior scientist named Lena Lin, and our mouse technician named Darwin Romero, who've been leading the COVID efforts to treat mice during this difficult time. On the clinical side, we have a team of, again, skilled and experienced clinicians led by Irene Manoli, Oleg Shalachkov, and Jennifer Sloan, who together with myself have been studying the natural history of these conditions since 2003 here at the NIH. And we're fortunate to have the support of the NIH and also all of our colleagues within the intramural research program, specifically yourself and your colleagues as well, Karsten. And it's been very exciting to work on the protocols with you and your team. I must say that and acknowledge the great contributions you have all made.
Even though we are at this point addressing quote unquote only 4 disorders, 2 for you, Chuck, 2 for us here at NDS, neuromuscular junction disorders and neuromuscular and neurometabolic disorders. But really what we do is meant to open up the field for many more of rare genetic disorders by making sure that the path to gene therapy is as smooth and efficient as it can possibly be. And so we are studying here disorders that we're particularly good at studying. We're setting ourselves up for success, but we have success, the field will have success, and the path for gene therapy for other rare disorders will be ever so much easier. That's really the goal and the hope for PaVe-GT.
You know, it's been amazing over this past year that despite the difficult times of COVID and the difficulties of conducting clinical and laboratory research, that the team has persisted. And we've made a lot of progress in the laboratory and in the clinic on the clinical side. And we continue to see patients remotely and work with our colleagues all around the NIH to develop protocols that we hope can form the basis of a broad range of treatment protocols for all children with metabolic disorders that might be treated using AAV.
Chuck, you're absolutely right. The COVID time was really a really tough time for us to try to continue with the research, but we have not stopped. The clinical team offline was working very hard on identifying the patient population, the outcome measures, planning ahead at this early time for clinical trials in the future, while the laboratory team was really sitting on hot coals to get back to the lab, to the experimentation, something that you can't do online. You have to be at the bench. Particular Janelle Hausam, an extremely talented postdoc who joined us from the University of Maryland specifically for PaVe-GT and developed gene therapy for the neuromuscular junction disorders, along with the postdoc student Eric Esposito. And they went back to the lab as soon as it was allowed, fully masked up, socially distanced, but getting experiments going, getting animals up and running, and developing the therapeutic vectors we plan to move forward to the clinic. And hopefully we'll ramp up to full 100% very soon.